Great to see everybody. I'm looking forward to the next hour, and I'm really pleased to be working with Liz on this. What we're hoping to do is to bring insights both from the lab-based tools and from the synchrotron around what you can do within X-ray imaging in life sciences. So welcome everybody, and I hope you enjoy the next hour. So my background is actually light microscopy rather than X-ray imaging. I come from a kind of confocal, super resolution, light sheet background, and I discovered X-rays in 2015. And what occurred to me at the time was the potential power that these had for complementing other microscopy technologies within life science. So my part of the talk today will be to introduce X-ray imaging to you, why it's valuable to life science research. We'll talk a little bit about samples, how you can prepare them, how you can contrast enhance them to get them ready for imaging using X-rays. And then delve a little bit into the kind of experiments that are possible, the different application spaces and the considerations associated with those. Liz will then talk about the similar applications that you can do at the synchrotron, or the extension of what you can achieve at the synchrotron. And I hope that the two halves of this will really complement each other. So we're not hoping to give you a completely comprehensive view of everything around X-rays, but really an introduction to dabble your toe in the water. And I hope by the end of it, it enthuses you to want to find out more about how X-rays can support you and your research questions. We have created a landing page on the ZEISS website for information surrounding this whole webinar series, so both from today and from the whole series of electron microscopy and X-ray microscopy talks. So that will consist of a number of different collateral. So over the next few weeks, if you want to visit the webpage via the QR code, then please feel free to do so. So let's start with the basics of why you would want to image using an X-ray system. So the huge value of X-ray imaging is that it's a non-destructive technology. You can acquire information from your sample without physically having to section the sample at all. And this is hugely powerful for a number of different applications. Here you can see that this is a mouse kidney that's been imaged using X-ray technology. It hasn't been sectioned. You can look at the internal structure without any problem at all. And you can then go on to look at that sample in more detail using other technologies because you haven't destroyed it. And actually, if you look over the number of publications that are coming out of life science in particular using X-ray technology, this is over the last 20 years or so, and I just pulled this from PubMed using a number of different search terms around X-ray imaging. There's been a huge uptick, particularly in the last 10 years, of research publications invoking X-ray technology. And this really shows a combination of things. It's the sample strategies and how you actually put these experiments together that have developed. It's also the technologies themselves that are showing themselves to be very powerful for providing different scientific insights. So it's an exciting time for the technology area, and it's also an exciting time for a number of different application spaces that are making use of this non-destructive technology. If we think about the areas where this application or this technology can really make a difference, the classic one within life sciences, I would say, would be around mineralized tissue. So bone and mineralized tissue are historically quite challenging to image using light microscopy. They're pretty dense. They can be sometimes quite challenging to section. So using a non-destructive technique to be able to look at internal structure in 3 dimensions has huge value and power. And the bone community in general use X-ray technology a huge amount to look at bone morphology and internal structure. But other areas of biology are now kind of keeping up in terms of the amount of usage of X-ray imaging, including developmental biology, so looking at things like mouse embryos, soft tissue and organs, and this can extend to tumors and cancer research. There's a raft of great work that's being done in plant sciences, so everything from seeds to the floral parts of plants. Then over to natural history. So you can't get more precious than a sample that is millions of years old. And the idea of being able to physically section it is completely unthinkable. So having a non-destructive way of being able to look at the internal structure of natural history specimens is incredibly valuable. Equally, looking at implants and tissue engineering. How does your tissue regenerate around your implant? What is the biocompatibility of your implant? These are great questions that X-ray imaging can address. Equally, within neuroscience, looking at neural structures, looking at brain specimens, there's a lot of exciting work happening in that area. And then when we look to being able to combine multiple technologies together, so if you're looking to go to 3D electron microscopy, for example, using a 3-dimensional map like you can achieve with X-ray imaging allows you to be able to locate with great precision exactly the region that you want to be able to acquire with 3D EM. So these are a few kind of key areas where there's been a lot of interest and an uptick in terms of the amount of work being done in these areas. And I hope over the next few slides, you can understand a little bit about why that is. So when I first came to X-ray imaging in 2015, as I say, I came from light microscopy, and one of the things that struck me most was the complexity of the nomenclature in this imaging space. There are so many different terms and abbreviations which seem to get used interchangeably with each other. And for me, particularly, understanding kind of the differences between what these terms mean, what the technologies are, which applies to which, was quite confusing. Are they all the same thing? Are they different? But the thing that helped me in terms of being able to find my way through that forest of words was that actually the basis of how all these images are generated, irrespective of the terminology associated, is the same. So tomography imaging, X-ray tomography, is the basis of all of those terms that I showed on the previous slide. And to actually acquire a tomography image, what you're doing is capturing a number of different two-dimensional images from the sample at a slightly different rotation. So here we have our mite, I think it is in this case, that's mounted inside the X-ray imaging system. That mite or bug is rotated through 360 degrees, and a number of different two-dimensional projection images is captured. So the way that it works inside the X-ray system, you have your source of X-rays, then you have your specimen, and then you have your detector. So you're looking at the transmission of X-rays through that specimen, and these are the two-dimensional transmission images that we're looking at here. You can collect hundreds and very often thousands of two-dimensional projection images by rotating that specimen and capturing those data sets. And you can then computationally generate a 3D render of that image from those two-dimensional projected images. So what we're seeing on the right-hand side here is the computationally reconstructed image of those two-dimensional projections. And the really great thing, if you've captured your data set through 360 degrees, coming from light microscopy, this was quite astonishing for me, where I'm used to in light microscopy having a better resolution in XY usually than in Z. Using a three-dimensional approach like X-ray tomography, when you've rotated the sample through 360 degrees, your resolution is the same irrespective of orientation. So you can computationally section your specimen through any way you wish. And this gives you a huge amount of power to explore kind of what's happening structurally within that specimen. So the basis of tomography is the same irrespective of the technology platform that you use. And to pick out a few of the technology platforms that you can use to be able to generate your image, if we start with the synchrotron, Liz will talk a lot more about what the synchrotron is in the second part of the talk. But essentially, it's a really, really very high flux source of X-rays. And that source of X-rays can be shaped as a parallel beam or a cone of X-ray beam. The X-rays pass through that specimen, and then optical magnification, so again, anyone used to a light microscope will recognize these objective lenses are used to be able to generate the increase in resolution or magnification. So you have a huge high flux of X-rays, which allows you to have a really high throughput of specimens at the synchrotron, but it's the objective lenses, which are coupled to a scintillator to turn your X-rays into visible light, that allows you to generate your magnification. If we move into the lab, we don't have access to such a high flux of X-rays. You don't have access to the synchrotron, so you use a different form of generating X-rays. And this is a microfocus X-ray source, which has a much lower flux, and the beam of X-rays is actually generated as a cone beam, as shown in this graphic here. And the way to generate an increase in resolution or increase in magnification using this approach is something called geometric magnification. So you physically move your source and specimen closer together. And this creates an increase in magnification that can be captured on the detector. And this approach is called geometric magnification. So there's a limit to how much magnification you can get with geometric magnification, because physically at some point your sample size becomes limiting. Or indeed, if you want to mount your sample inside something like an in situ rig, or perhaps a temperature controlled device of some sort. So you're limited in terms of how far you can push the magnification. So if you want to be able to go even further in terms of resolution, that's where using a combination of optical and geometric magnification can come in really useful. So the X-ray microscope is a combination of geometric magnification. You're still a lab-based system. You still have a microfocus source. So you can physically move your sample closer to the source to get higher resolution. You can also combine that with the optics and get optical magnification as well. So you simply change your objective lens like you would do on a light microscope. And that gives you access to higher resolution information and some really stunning data, which I hope I can show you over the next few slides. So it's this combination that really sets it apart. But X-ray microscopy and micro-CT are actually only part of a whole breadth of different imaging technologies in X-ray imaging. So on the left-hand side here, we have the clinical CT scanner. So some of you may have actually been inside one of these, but they can be used for imaging human limbs. In a lab-based system, you could use them for the in vivo micro-CT, you can use for imaging small rodents. You could use rodent bones within a micro-CT system. You can use small rodent bones or pieces of bone using X-ray microscopy. And then pushing the resolution stages even further, you can move to the synchrotron with nano-CT. This is a really nice figure taken from a paper that was published earlier this year, looking specifically at the technology for bone. But I will emphasize here, it isn't just about bone. There's so much exciting stuff happening in addition to the world of bone in life science research. The same paper had this graphic, which turned the technology platforms into actually what you'd be able to achieve in terms of imaging. So on the left-hand side here, clinical scanning. So these are human bones that have been imaged here. In vivo, you can look at what's happening in the live animal. You can then move to micro-CT to start looking at the resolution of the bone morphometry inside the bone, X-ray microscopy if you need that higher resolution for the structures that you're looking at, and then moving on to the synchrotron for all sorts of other exciting experiments. So it's this real range of technology across multiple scales and capabilities that makes this area really very exciting. So how can you actually generate the contrast you need in your sample to be able to get this three-dimensional structural view of your specimen? Well, one approach is called absorption contrast, and absorption contrast essentially shows the differences between structures based on their X-ray absorption. So as your X-rays pass through the specimen, some parts of the specimen will absorb more X-rays than others, and therefore that will be reflected by what the detector can see. So there may be natural differences in your specimen. It may be that you have this X-ray absorption difference already, in which case you don't need to do too much to your specimen, or you may need to generate additional contrast using contrast enhancing agents. So it depends really on what the experiment is. So this is the kidney specimen again that I showed at the beginning. And if you look at the cross section where that yellow line was, this shows you that you can view that image from all sorts of different angles, all at really high resolution using this contrast enhancement. But for some specimens, it's not possible or desirable to be able to use contrast enhancement. Indeed, you may not actually want to use absorption contrast at all. And in that case, there are alternative means of being able to generate contrast. So this is a very example of that. This is a fly or a bug that's in amber. So it's not possible to add any absorption contrast agent at all. But what does exist in this specimen is a difference in X-ray refraction between different components of the specimen. So the membranes of the specimen have a different refractive index to the amber, and it's possible to use something called phase contrast to be able to accentuate that difference. So in this graphic on the left-hand side here, if we imagine that's the fly, at the edge of the fly or the edge of the membrane, you have a difference in refractive index between the amber and the wing. And that causes a slight refraction in the X-ray beam, which means it lands on a different part of the detector. So you can see this difference in the specimen. And when we actually look at that specimen in phase contrast mode, you can see how that looks. So it really generates this huge contrast at the edges of the specimen. So it's a combination of absorption contrast, which is what we see on the right-hand side here, and absorption plus phase contrast that enables you to be able to visualize structures that you wouldn't otherwise be able to visualize. So it's not just about absorption contrast. Very often it's about phase contrast. There's super exciting stuff happening with phase contrast at the synchrotrons as well. And it's important to be aware that it's not just about generating contrast in terms of density differences. These are a few examples of where phase contrast or propagation phase contrast has been used. So in the top left-hand corner here, Liz was joking when I was talking to her yesterday, someone gave up their lunch for this experiment. This is actually a pear that you eat. And what you can see here is that the cell walls are highlighted because there's a difference in X-ray refraction index between the pear and the cell wall and the other components of the cell. And it allows you to be able to pick out those differences, generate that contrast in the image, and then segment out the whole network within that pear specimen. Equally, the image on the right-hand side here, so the absorption contrast, again, this is another example of a fly in amber. With absorption contrast only, you can't really see much of the specimen, but as soon as you look at absorption contrast with the propagation phase contrast, it really makes that specimen pop, and you can see those interfaces beautifully well. Final example of that at the bottom here is a publication from 2015, where they were looking at the structural properties of this wrapped carotid artery. So they really didn't want to include any contrast enhancement here because it would impact on actually the structural integrity of the very structures they were looking to capture. So they used propagation phase contrast to pick out the membranes, and this gave them enough detail that they could actually segment out the different layers of the carotid artery, which gave them access to the information they needed for their study. So moving on a little bit to the sample preparation. I think anybody who's worked in microscopy knows this to be true, but particularly for X-ray imaging, you can't get good data from a poorly prepared sample. It really needs to be well prepared, it needs to be well secured, it needs to be well mounted to be able to generate the best possible results. And for X-ray imaging, there are three main considerations that you need to take into account. You need to make sure that you have sufficient contrast enhancement. So if you're using absorption contrast, is there enough difference between your different components to show well in the image? How are you going to mount that specimen? Will it be in liquid? Will it be in air? Will it be mounted inside some kind of a resin? And then how are you going to keep it still? So it's absolutely critical for X-ray imaging that as you turn your sample through 360 degrees, the specimen itself, apart from the rotation, does not move. It's very important that's the case. If we start with the first one, so generating contrast, one of the absolute gurus in the field for this is Brian Mechner. So he published a paper in 2009, which has been extremely well cited, looking at the kind of different stains you could use for enhancing contrast in biological specimens. And I would encourage you to visit that paper and his homepage to give you a bit of inspiration about the possibilities, particularly within soft tissue, for being able to generate contrast in the structures that you're interested in. Iodine, PTA, osmium are all very common contrast enhancement agents that you can use. But yeah, those references are a great starting point if you're interested in setting off down the road of X-ray imaging. Equally, if you're looking at being able to combine different protocols together, so the volume community has a few very established now protocols for being able to prepare their specimens, and some of these work really well with X-ray imaging too. So this osmium prep that's commonly used for serial block phase SEM also works really well for X-ray imaging. And what you can see here, this is an example taken from the Crick. This particular group were really interested in identifying the carotid body, and they wanted to be able to acquire that at much higher resolution using serial block phase. So they took this specimen, imaged it in the XRM, identified the region they were interested in, and then moved it over to the serial block phase system. Another fabulous resource, if you're interested to know more about this workflow, the Crick guys put together a great workflow video, which takes you from the confocal, so the LSM, through the X-ray system onto the electron microscopes, taking you step by step through what's involved with the technology and what you need to do staining wise. So I would definitely recommend, if you're interested to know more, to visit that video for yourselves. But if you're not actually wanting to do any contrast enhancement at all, there are many other options too. So critical point drying is another really effective way of being able to generate contrast in the image. This is a Drosophila fruit fly that's been critical point dried and then imaged using the X-ray system. And you can see there's great access to all the internal organs there that you can segment out. And equally, corrosion cast imaging, so you can flood your structure with a liquid, which then solidifies. You can then remove the tissue from that and image your specimen from the corrosion cast. There are lots and lots of different possible options, and it's a question of looking at what other people have done and deciding what the best route would be for your specimen. So once you've got the right level of contrast enhancement, you're pretty confident that the actual imaging will work itself. Next thing you need to do is actually mount the specimen in the system. So this is not the in vivo stuff. This is samples that are fixed, so you're going to keep them still for the duration of the image acquisition. And these are just a few examples. I've seen some crazy setups in my time: Lego, polystyrene, blue tack, sellotape, all sorts of things involved with being able to keep the specimen still. Well, but these are a few examples. So on the left-hand side, we have a resin-embedded section on a plastic slide, and this is taken from that video I mentioned from the crick. So it was glued onto the pin to keep it still. The next one in from the left is actually imaged in ethanol. So this was a tumour in this pipette tip that's been sealed either end with gum to ensure that it doesn't have any evaporation. So you can image in liquid too. You're not restricted with X-ray imaging, you can image in liquid as well. In the middle, we have the piece of the amber. So this is the fly in amber that's been stuck, super glued to the pin to keep it still in place. I will show on the next slide some amazing stuff that's been done with plants. But the great thing about plants is you can actually grow them inside the system. So this is a plant that's actually in a tube in soil. So it's being imaged live. And then on the right-hand side, this is a heart that's been bedded into low melting point agarose into a falcon tube and imaged that way. So there are many, many different options, but the key thing is you need to keep it still for the whole duration of the image acquisition. So now for a few examples, and if I start with the plant example that I showed on the previous slide, to give you an example of the technology, here you have your sample that's mounted here. This is your X-ray source, and this is your flat panel detector in this case. So this kind of level of resolution gives you access to the soil. So you can see the different components of the soil. And then down here, you'll begin to be able to make out the root. So you can look at the network of the root, where it grows, how it branches, those kind of questions. Once you've identified the root and the location of the root, you can then switch up your resolution and move over to a higher resolution image. So this is with the 4x objective lens now. So here we've switched to the objective lens that allows you access to high resolution information. And that enables you to be able to reach here. These are individual fungus spores, which Keith managed to segment out from his specimen. So it really depends what kind of information you need as to what resolution you would go for. Equally here, we have the axillary buds. So these have all been imaged without physically sectioning the specimen. I keep emphasizing that. The wonderful thing about X-ray imaging is that it's non-destructive. And the volume of information that you can capture using this kind of technique is just breathtaking. So you have access to sections and sections and sections and sections of data. So you have your information, but you have it in the context of the 3D structure. And very often that's the key differentiator for X-ray data, is that you don't just see your structure of interest, you see your structure of interest in the context of everything else. So it gives you access to incredible data sets like this 3D one from Keith. So multiscale imaging isn't just applicable in things like plants, it's also applicable in things like bones. This is a bear jaw. It was actually taken from the bear before it was imaged. But what we see here is that the group were interested in the interface between the bone and the jaw, the tooth and the jaw, sorry. So they imaged at relatively low resolution to get the whole overview of the jaw. You could then zoom in on the particular tooth. And once you've identified the particular tooth of interest, you can then look at the interface between the tooth and the jawbone. So this multi-scale approach, again, if you've used a light microscope, it probably feels really familiar. You just switch the objective lens and go up in magnification. And it's the same principle. In X-ray imaging, you're just working constantly in 3D. When you start to push the resolution even further, so this is the paper from earlier this year, looking at pushing what you can achieve in bone. Looking at osteocyte lacunae is something that's been done in terms of bone structure for a while, but the additional contrast that you can now reach, perhaps even showing remnant calcified cartilage. So here you can see slight differences in contrast in the bone. And that holds a lot of excitement about what this could possibly mean for bone biology. So this technology area is definitely moving and there are new things to be found. Possibly one of my favorite data sets. This is a mouse embryo that showcases some of the detail that you can achieve for being able to compare things like genetic models. So it might be that you're looking at toxicology studies, you might be looking at genetic models, you might be looking at the effect of certain things on different organs within the embryo. This is a mouse embryo. On the right-hand side, you've got the three-dimensional reconstruction, and the images on the stacks that you're seeing on the left and in the middle are different views of the same data set, again showcasing how you get isometric voxels. You get the same resolution irrespective of which orientation you're looking at your specimen at. the access to all of that information inside the embryo gives you so many possibilities for looking at the effect of different things. Next example, this is an organ now. So this is a mouse embryo heart. So it's a 1.5 to 2 millimeter size specimen. And the goal of this study was to detect these pretty complicated cardiac malformations. So they were looking at differences between different embryo hearts. These are single 2D slices through the 3D data sets, and the 3D renders you can see here. But again, having the resolution and the contrast in that specimen gave them possibilities to be able to spot these differences pretty efficiently and effectively for their study. Looking to push the resolution even further, so we've gone from looking at whole animals and whole organs right through now to looking at single cells. These cells have been prepared for 3D EM. They are embedded in resin and they are osmoneuronal acetate lead stained, so that the classic 3D EM protocol. What's amazing about this is you can actually get to subcellular resolution. So we've gone from whole organs right down to subcellular resolution. So the flexibility of this kind of X-ray imaging approach gives you so many different options. You can then go on and image this with electron microscopy if you wish, but if this kind of resolution answers your question, then this can be the endpoint in 3D. And this is just a single 2D section or two single 2D sections through your 3D data set. Here, we also have another example of a 3D EM data set. Finding the sample was key here. So locating the region of interest, then chopping down the specimen to the particular size. So the guys at the crick here, this is the tissue of interest. They've actually measured on their specimen, cut to size, and then subsequently gone on and imaged that using the seroblock face SEM. So it really sped up the rate at which they could achieve this workflow, being able to identify very clearly exactly the region that they were interested in imaging before they went on to 3D EM was incredibly useful. Another example of a correlative workflow, this time within neuroscience. So this is a Drosophila brain now that's actually has genetically, the neurons of interest have been genetically tagged and genetically labeled, and they're shown in green. So this is the X-ray data set that's now been segmented out with the neurons of interest in green. And the goal here was to be able to capture at high resolution using the focused ion beam SEM, those neurons of interest. So identifying them, first of all, getting the genetic stain to work well is not always straightforward. So there were a few brains in this study that just didn't work in terms of staining prowess. But rather than finding that out at the focused ion beam stage, we were able to identify that at the X-ray microscope stage. So it saved a huge amount of tool time on the focused ion beam SEM side of things. But now, once you'd found with the X-ray imaging approach, the samples that had worked well, where the staining had worked well, it was possible to then take them over to the focused ion beam system, capture the high resolution data set in a pretty straightforward way because you knew exactly where it was. And you had all of that information in the context of the Drosophila brain as a whole. So as an experimental workflow, combining these different technology areas together really holds a huge amount of value. And my final example here, this time is of a moth. So this is a moth abdomen where these were particularly interested in the reproductive organs of this moth. So the goal here was to be able to capture the reproductive organs at high resolution. And as I understand it, that's important for classification of moths. I'm not an expert in this field. But the digital documentation of this moth specimen really was a small part of a much larger project to be able to have digital documentation of the specimens in general at the museum. So non-destructive determination and classification was really important for them here. This specimen was also mounted on a metal pin, as many museum specimens are. So that causes its own challenges when it comes to actually being able to capture the data sets using X-ray imaging. So that's the end of my part. I hope I've given you at least a few insights into the possibilities for X-ray imaging and how the technology works. And I'm now going to hand over to Liz. So we've talked a lot about the lab tools, and Liz is now going to introduce the possibilities and, frankly, incredible imaging options that are now available at the synchrotron.
Welcome again, everyone, and thank you very much for listening to us talking about X-ray imaging. As Gleb said right at the beginning, I Liz Duke, I recently joined EMBL to develop hard X-ray imaging techniques for soft biological tissue. And prior to that, I was doing cryosoft X-ray microscopy for imaging single cells. And I have to admit that I do have a shameless plug here. I do have postdoc and PhD student opportunities. Should anyone be interested in all the exciting opportunities that X-ray imaging provides, then please do get in touch. Or if you know anyone who might be interested, please pass on the information to them. And so I will just start by briefly introducing synchrotrons. I apologize if all of this is well known to you, but I decided it was easier to explain what's going on at a synchrotron rather than assume everyone knows. So as Rosy highlighted, synchrotrons are an incredibly powerful source of X-rays. They're big facilities, so they're the sort of facilities that a country might have one or maybe two. They're often multinational facilities. This image here on the left-hand side is Diamond Light Source, which is the UK's national synchrotron in just south of Oxford in the UK. But so the X-rays are generated by circulating electrons at the speed of light around around a circular ring, and the result of the electrons being accelerated around the ring, they generate X-rays naturally, and these X-rays are effectively funneled off down beam lines that go off at a tangent. And because the electrons are going round and round and round the synchrotron the whole time, all these beam lines can operate simultaneously. And what you find at synchrotrons is that beam lines are built and operated with one particular technique in mind. So you'll probably have heard a lot about macromolecular crystallography and the use of synchrotrons for structural biology, and so most synchrotrons have structural biology beamlines. There are also spectroscopy beamlines where you can do elemental analysis, and also there's the opportunity to use your synchrotron to do imaging. And so I will focus on what we can do using X-rays to image. As Rosy commented, there are many, many different forms of X-ray imaging. I'm not going to cover them all because otherwise you'd be here till next year, and I think you probably all have better things to do. But one thing I would highlight is that fundamentally every microscope is the same, be that an electron microscope, a visible light microscope, or an X-ray microscope. They're all fundamentally the same. They all require some form of source of particles, be they X-rays, visible light photons, or electrons. They then need to have some way of focusing those electrons, X-rays, visible light photons down onto the sample, and then collecting the X-rays, and then those X-rays are then focused onto a detector. And you need to choose your source of X-rays properly. You need to have the right optics to focus the X-rays onto the sample, and then you need to make sure the sample is appropriate, and then also the detector is appropriate. But fundamentally, all microscopes are the same. They all have some form of source, some form of optics for the sample, and an appropriate detector. And so with that in mind, I'm going to talk a little bit about cryo-soft X-ray microscopy, which is using soft X-rays. And this technique is really only possible at synchrotrons in any routine way. You can get laboratory-based soft X-ray systems, but they're not routinely available. And so we really do need the synchrotron to do cryo-soft X-ray microscopy. And this is an illustration, still at Diamond Light Source, of beamline B24, and I would strongly encourage you to go to B24's website, where they have amazing videos of all the data that you can collect and showing you how to prepare the samples with links to their YouTube videos. So really do encourage you to go and look at that, because also what's really powerful about cryosoft X-ray microscopy is the ability to correlate the X-ray results with cryo super resolution or cryo conventional resolution fluorescence microscopy. And the combination or the correlation of these two imaging modalities is incredibly powerful. But back to the beamline itself, if you remember from my previous slide, I was talking about how all microscopes are the same. You need some form of X-rays. Well, here in this slide, our source of X-rays basically starts from the bending magnet and then through the optics that then focus the X-rays onto the exit slit. So all of this up to the exit slits is your X-ray source. So actually, as far as a user is concerned, you don't really need to worry about anything at this point. So we have our source of X-rays here. It emerges from these slits here. And then we have our sample here, and we have a capillary condenser that focuses the X-rays down onto the sample. The zone plate collects the X-rays once they've passed through the sample and are going on to the detector. So fundamentally, it's just a microscope. It just happens to be quite an expensive one, but it's still just a microscope nonetheless. So what is cryosoft X-ray microscopy? And we use the term soft to distinguish these X-rays. They're very low energy X-rays. They're only 500 electron volts, and the reason why we use them at 500 electron volts is that what they allow us to do is the X-rays pass through the sample, and between the carbon K edge, which is here, and the oxygen K edge, you have preferential absorption of the X-rays by the carbon-containing proteins versus the oxygen-containing water in the surrounding areas. And so, but you also have the penetration power of the X-rays as well. And I tend to look at cells as if they're fried eggs. You have the nucleus, usually in the center, and surrounded by cytoplasm. And what we have is the ability for the X-rays to pass all the way through the cell, including the cytoplasm and the nucleus. And that's in contrast with electron techniques, which are limited to either going through the periphery of the cell, or you have to shave off bits of the cell in order to make the cell much thinner, so you can then image it with electrons. The X-rays can penetrate very nicely and penetrate all the way through. But what we do have to do for this technique is plunge freeze our cells in order to protect them from the heating effects of the soft X-ray beam. And also, because the X-rays are very easily scattered, for this data collection, for this type of X-ray microscopy, all of our data are collected in vacuum. And so this is just the inside of the X-ray microscope. Our samples, the cells are grown typically on 3mm electron microscopy grids, and these are mounted inside the vacuum chamber on this mount here. Here is the optics bringing the X-rays down onto the sample, and here is the support for the optics that will then collect the X-rays having passed through the sample to focus them onto the detector. To give you an idea of scale, this here is holding a 3mm grid, and this distance here between the sample and the zone plate objective is about 1.5 millimeters. So everything is very, very, very small. But what sort of data, what sort of information can you get? And this is just a 2D slice from the three-dimensional data showing you that what you're seeing is the carbon dense features in the cell, so that's anything that's particularly lipidic, is really shows up very, very nicely because of the fact that the X-rays are absorbed by the carbon more than the surrounding oxygen, watery type features. And so you could see, for example, the mitochondria and the cristae inside that, and other features. And again, I would urge you to look at B24's website, where they show some absolutely stunning data, looking at virus entry in cells, and huge opportunities to study intact cells in their native state, where the only sample preparation that's been done is plunge freezing. But moving on to high energy X-ray imaging, which is what the area I'm working in now. And over the last 10 years, there's been some amazing tour de force publications coming out, sort of starting with the Julian Moosman's work here in Hamburg, looking at frog egg gastrulation, the work from the Swiss light source, looking at the fly wing beating. And then also what's been amazing over the last few years is the interest in using X-ray imaging to look at brain and to look also at other soft tissue. And the information that's available there is quite considerable, and also particularly the work of Aaron Kuan in a collaboration with Alexandra Pacureanu from the SRF is then correlating light microscopy, volume electron microscopy, and very high resolution X-ray microscopy techniques. It's incredibly powerful. The area of X-ray imaging that I have a particular interest in is using X-ray imaging to bridge the gap between the very high resolution electron microscopy techniques and particularly the fluorescence microscopy confocal light sheet type techniques in a correlative approach. And that's an area that I am very, very interested in. And so I'm looking at exploiting the X-rays to image what I would call bulk samples of tissues. So that tends to be the sort of biopsy, 1 millimeter biopsy punches and similar things. But we can also image small organisms and organoids as well. There are many possibilities. So the beamline we're using here in Hamburg is, in fact, we're repurposing a macromolecular crystallography beamline. So this is the EMBL's macromolecular crystallography beamline P14 here at Petra 3. And what we have here is a really nice uniform top hat beam, which is very, very stable, which is essential for good imaging. But we're also able to take advantage of all the technology that has been developed for macromolecular crystallography. And that's particularly a very nice diffractometer, but also all of the automation. And as Rosy alluded to in her introductory first half of this session, where we have huge advantages in the synchrotron is the fact that we can do everything much quicker because we have very, very intense X-ray beams. And so by including the automation, it means we can actually have an even greater throughput for large volumes of samples by using the robotic sample changer. And so here in Hamburg, we're developing what I'm calling HIT, which is high throughput tomography. It's phase contrast tomography. We collect data at 4 distances, and it takes us 5 minutes to mount a sample, collect all of those data sets, those 4 data sets, and then dismount the sample. And then there is a pipeline developed for processing the data. And so very, very quickly, we can generate huge volumes of data, which in itself is an enormous challenge. But the opportunities are incredible. And so this data, in contrast to the soft X-ray data collection I mentioned earlier, all of these data are collected at room temperature and in air. We are currently using 12.6 kV, but at the minute, again, all these techniques are so new, we're exploring what energy of X-rays to use. And we're also exploring the sample mounting, the sample preparation protocols required. It's very nice. We can use a lot of the volume electron microscopy, heavy metal staining, resin embedding protocols, but we're also working with histology samples, which are paraffin embedded. And in fact, we've got some beautiful data from samples where we've used no staining at all. So it's the phase contrast that you get simply from the structures within the tissue is incredible. And what we're able to use is the spine pin that allows us to then interface with the robot system so that we can then get through large quantities of samples. And so this just highlights the fact that we are using the robot, and any crystallographers or people familiar with crystallography will recognize the pucks and the pins. And here we have a Platinarius dumerilli sample mounted on the end of a pin. And in fact, this was in the very, very early days of this technique, where we just were mounting everything on a block of resin, as opposed to actually mounting it on the end of the pin, which is what we're now doing. And so just to give you a few examples of what we're doing. And so this is Platinarius dumerilli. It's 6 days post-fertilization. And these data we collected these data, and then Jonas, my colleague, then was able to analyze the data. So this is heavy metal stain. So this organism is a very well-studied, well-characterized organism for developmental biologists, but also there's opportunities to look then with high numbers of samples, looking at environmental effects on the organism and such like. And so this is just an example of what you can do. And so these data were collected in 5 minutes on the P14 beamline just before Christmas. And so you're able to see subcellular resolution, what they've been able to do with these data is then find areas of interest that they can then target for even higher resolution with volume electron microscopy techniques in Heidelberg. And so moving on then to a totally different type of sample. So this is lung, courtesy of the University of Gottingen. And this is paraffin embedded but unstained. And again, so this is a 1 millimeter punch biopsy, and we're able to see the internal structure of the lung very, very beautifully. And the lung is a really, really nice organ to study because it basically is full of pockets of air. So you have very, very nice, naturally occurring changes in phase as the X-rays pass from tissue to air pockets, back to tissue, except of course it's not air pockets, it's the paraffin, but you do have that change in phase, which helps. And so then moving on, this is brain, courtesy of the collaboration we have with the Francis Crick Institute, and particularly Carlos in Andreas Schaefer's group. And so this is heavy metal stained, resin embedded brain from a mouse. And here we're able to see the different sections of the brain, and we're down at the level in this particular sample where we can see the apical dendrites. And so the opportunities there to use this X-ray imaging again as a means of targeting areas of interest in bulk samples to allow you to collect much higher resolution data by other techniques is there. And so one of the challenges, as that I alluded to, is the fact that we're generating huge volumes of data in these data sets, and keeping track of them and being able to handle them is a huge challenge, which we are slowly getting to grips with, but it is certainly a challenge for our collaborators who are less used to dealing with the huge volumes of data. And one of our goals is to make our data as easily accessible as possible, and making sure that we keep track of all the information. And so being located within the EMBL means that we do have access to all of the databases and such like based at the EBI in Cambridge, which is great. And with that, I will finish, and just to highlight the huge power we have of X-ray imaging that allows us to find the images, find the needle in the haystack. And I would like to very much thank you for your attention. I hope Rosy and I have given you a flavour of the huge opportunities with X-ray imaging. We've only scratched the surface, and I'm sure that we can try and answer some questions if you have any. Thank you very much.